PHARMA Pharma interview question for QC Part 1. Basic interview question. First question is difference between the LOD and water content. LOD. LOD means loss of drying. LOD measures the weight loss of sample when it is dried under controlled conditions, typically using heat. The weight loss is attributed to the evaporation of moisture and volatile compounds. Water content. Water content, also known as moisture content, measure the amount of water present in a sample. This can be determined using various methods, including Carr-Fisher titration. Key difference. First, purpose. LOD measure weight loss due to drying, while water content measures the actual amount of water present in sample. Second, methodology. LOD involves drying the sample, while water content can be determined using the various methods. Third, results. LOD results are typically expressed as a percentage of weight loss, while water content results are expressed as a percentage of water present in sample. Fourth, interpretation. LOD results may include the loss of volatile compound, while water content results specifically measure the water present in a sample. Second question. What is a ICH? The International Council for Harmonizations developed guidelines to ensure the safety, quality, and efficacy of pharmaceutical globally. What is the method validation? Method validation is a documented evidence that the test procedure is suitable for intended purpose. Fourth question. What are the different type of apparatus listed in ASP? First type is basket, second paddle, third reciprocating cylinder, fourth flow through cell, 5 paddle over disc, 6 rotating cylinder, 7 reciprocating disc. What is the principle of UV spectroscopy? It is the based on Beer's and Lambert's law. Monochromatic light is passed through in absorption of solution, decrease the intensity of light in relation to absorption solution and concentration of solution. What is the range of UV and visible radiation? UV range is 200 to 400 nm. Visible range is 400 nm to 800 nm. What is the quantitative and qualitative chemical analysis? Qualitative chemical analysis, first of all definitions, identify the presence or absence of particular substance or element in a sample. Objective, determine the chemical composition of sample. Method, chemical test, precipitation, colorimetry, chromatography, example paper chromatography, thin layer chromatography. Spectroscopy, infrared, nuclear magnetic resonance. Example, identifying ion in a solution, sodium chloride. Detection the presence of specific compound. Example, glucose, amino acids. Quantitative chemical analysis. Definition, determine the amount or concentration of a particular substance or element in a sample. Objective, Measure the quantity of substance or element in a sample. Methods: gravimetry, weighing, volumetric, example titration, spectroscopy, example automatic absorption, fluorescence, chromatography, example gas chromatography and liquid chromatography. Example: measure the concentration of glucose in blood, determine the amount of lead in water, quantifying the composition of mixture. Example: percentage of component. Key difference between quantitative and qualitative chemical analysis. Purpose: qualitative analysis identify the substance, while quantitative analysis measure the amount. Method: qualitative method focus on detection, while quantitative method focus on measurement. Results: qualitative results are typically yes or no or present or absent, while quantitative results are numerical value. What is normality and molarity? Normality definition: normality is a measure of the concentration of solution expressed as the number of equivalent weight of a solute per liter of solution. Unit: normality is expressed in unit of equivalent per liter or N. Equivalent weight: the equivalent weight of substance, its molecular weight divided by its valency, number of electrons transferred in a reaction. Calculation: normality is calculated using the formula N is equal to number of equivalent of solute divided by liter of solution. Molarity definition: molarity is a measure of the concentration of a solution expressed as the number of mole of a solute per liter of solutions. Unit: molarity is expressed in units of mole per liter or m. Mole: a mole is a unit of measurement that represents 6.022 into 10 raised to 23 particles, atoms or molecules. Calculation: molarity is calculated using the formula m is equal to number of moles of solute per divided by liter of solutions. Key difference between normality and molarity. Normality is based on equivalent weight, while molarity is based on moles. Normality is used for reaction where the number of equivalent is important. Acid base reaction, while molarity is used for reaction where the number of moles is important. Normality is often used in titration, while molarity is often used in synthesis reaction. Difference between disintegration and dissolution. Disintegration: if its time required for tablets and capsule to break into fragment a given set of conditions. Result recorded on time. Dissolution: it is the time required for tablets and capsule to release inactive ingredients in dissolution media given set of condition. In dissolution test percentage release. Result recorded on percentage. Difference between reference standard and working standard. Reference standard: exceptional high purity, often exceed the 99.9%, minimize the margin of error for comparison with the other materials. Characterization: rigorously characterized through the extensive testing determining the exact chemical and physical properties. Example: structure, melting point, IR, purity, etc. Serve as a definitive reference point for comparison. Source obtained from the reputed external sources, example pharmacopoeia, EVSC, BP. Certified reference material providers ensure the objectivity and consistency across the different laboratories. Use serve as the ultimate benchmark for quality control. Use for calibration of analytical instrument, validation of analytical methods, ensuring the accuracy, precision and reliability. Quality control testing, verifying the drug substance or product meets specifications. Calibration, not routinely calibrated, maintain its exceptional purity due to limited use, sets the gold standard for purity and characterization, provides the ultimate authority for calibrating and verifying the working standard. Analogy, master key for accurate analysis define the definitive properties of a material. Working standard, purity, good purity, typically above 95%, meets the specific quality control requirement for a routine analysis. Characterization, validate in-house to ensure the reliable result for specific uses may not require the same level of detail as a reference standard. Source, derived from reference standard, often prepared by dilution or calibration of a reference standard, more readily available for frequent use within a lab. Uses, daily calibration of analytical instrument, maintaining the accuracy without depleting reference standard. Intermediate checks and in-process control during the manufacturing cost effect the alternative for frequenting testing. Calibration, require regular calibration against the reference standard, ensure the accuracy is not compromised through use or storage. Frequency of calibration depend on specific use and stability of the working standard. Analog, key copy used for frequently open the door for routine analysis, require the verification with the master key to ensure the continuous accuracy. Thank you watching for Pharma Like and subscribe.